Hello and welcome back to Mr. Rowe Science. Today I'm going to talk you through the first part of your specification for GCSE about eukaryotic and prokaryotic cells within the cell structure part of the course. What you've got on the screen at the moment is the objectives for this part of the course. So you've got to know that the plants and the animal cells are the ones which are eukaryotic cells. They've got a membrane, they've got cytoplasm, and their genetic material is enclosed within a nucleus. Bacteria are more typical of prokaryotic cells. They're much smaller in comparison. In fact, bacteria are quite often smaller than a single organelle in a eukaryotic cell. They've got cytoplasm and the cell membrane still. This time it's always surrounded by a cell wall. The genetic material is not in a nucleus. Instead it's found in DNA loops, or it's found floating around in the cytoplasm in little rings called plasmids. And you also, for this part of the course, need to be able to talk about and do basic calculations for different magnitudes and including the use of standard form. So for a couple of eukaryotic cells, your animal cell looks like this. So you've got your cell membrane around the outside. That's going to control the things that go in and come out from the cell. You've got your nucleus, which is going to contain the genetic material. So that's your DNA that you find in eukaryotes. A few of the other things you've got in the cytoplasm is you've got mitochondria. So those is where aerobic respiration is going to take place to release energy for the cell. You've also got really tiny ribosomes, and these ribosomes are the protein factories of the cell. And for chemical reactions to happen, you've got your cytoplasm, which is going to be the jelly-like substance which is found elsewhere in the cell. For your plant cell, this is another example of a eukaryote, but this time they've got a cell wall outside of the cell membrane. The cell wall is made of cellulose, and it gives it additional strength so that it doesn't break when you've got water moving in or out and osmotic pressure. Inside of that, you've got lots of the same organelles as in the animal cell. You've still got a nucleus which contains DNA or genetic material. And plants are going to respire as well, so they're also going to have mitochondria. They also need to make their own proteins, so they're going to have ribosomes, just the same as you get in the animal eukaryotic cell. They also have cytoplasm, which is going to allow them to carry out chemical reactions, the same as the animal cell. They've also got a couple of extra things though. They've got chloroplasts, which have a dark green colour when you look at them down a microscope, and those are organs which are specially adapted for photosynthesis. As well as those, you've got a vacuole. So these vacuoles are there permanently in plant cells, and the job of the vacuole is it stores water and cell sap. Okay, there we go, so the vacuole is the final organ. So to highlight the key differences between these two, the things that the plant has that the animal doesn't is the vacuole, the chloroplasts, and the cellulose cell wall. Other than that, all the structures are the same because they're both examples of eukaryotic cells. For a prokaryotic cell, a bacteria would look a little bit more like this. So you get different types, but quite often they're either rod-shaped or circular. They've got a cell wall around the outside. Inside of that they've got a membrane. They've got cytoplasm, again for chemical reactions to take place. And within the cytoplasm, instead of having a nucleus, prokaryotes are going to have their DNA in a couple of places. They've got a large DNA loop, which is sometimes called a bacterial chromosome. And they've also got little loops of DNA floating around which are called plasmids. As well as that, lots of bacteria need to move around because they're unicellular, they're not part of a larger organism. So they have an organ called a flagellum, which looks like a tail, and it allows them to swim around and be motile, we'd call that. So those are your different types of cell. You've got your bacteria, which is a prokaryote, and you've got your animal cell, your plant cell, which are both eukaryotes. And main differences are the eukaryotes have all of their DNA found inside a nucleus, whereas the bacteria, or the prokaryotes, have it outside of the nucleus. If you remember from earlier, the last thing on this part of the specification is talking about different magnitudes and orders of scale using standard form. So the main orders of magnitude we work with are on a scale like this. So you've got things which are in whole metres, so that's going to be things like entire organisms. So like a human, you'd measure on a scale of metres. Something large and multicellular like us. Then what we're going to do for each step is we're going to get 1,000 times smaller. So if you get 1,000 times smaller than metre, you get a millimetre. And with a millimetre, that's going to be generally things like tissues are going to be 1 millimetre scale. You might get small groups of cells which are within that range. If you then get 1,000 times smaller again, you end up with micrometers, which has this funny U symbol. Micrometers are the usual unit you'd use to measure the size of most eukaryotic cells. So they're about 30 micrometers large would be a typical size for a eukaryotic cell. If we then get 1,000 times smaller again, then you've got nanometers. And nanometers are now getting really, really tiny. Things like viruses would be found on a nanometer scale. And finally, to convert between these units, which you may need to do, for example, when you're doing a microscope calculation, is as you go down each step of 1,000, you're going from 1 to 1 times 10 to the minus 3, 1 times 10 to the minus 6, 1 times 10 to the minus 9. So we're getting 1,000 times smaller each time. So if we have 1 metre, if we make it into smaller units, we times it by 1,000. So 1 metre is 1,000 millimetres. It's going to be 1,000,000 micrometers, and it's going to be 1,000 million nanometers, or 1 trillion nanometers. Okay, so with these, we could also write them in standard form, so that would be the notation on this side, whereas writing out the numbers in full would be over on that side. Likewise, if we've got a unit which is in nanometers, if we're going up and making it into the larger units, we need to divide by the number, divide each one by 1,000 as we're going up each step. So to go from nanometers to micrometers, the same measurement will be 1,000 times smaller. The only thing which sometimes confuses people is when centimeters are thrown into this, because everything works nice and neatly in gaps of 1,000, but centimeters are in 1 times 10 to the minus 2 meters. So you've divided by 100 rather than 1,000. So a key thing you can do to get around that is every time you're given a unit in centimeters, first of all convert it to millimeters first. So you just divide it by 1,000 and start off with the unit in millimeters to avoid getting confused later on. Okay, so there's what we've covered this time. So we've looked at eukaryotic cells, prokaryotic cells, their features and the differences between them. We've looked at plant and animal cells, and we've also done a little bit on scale and use and magnification calculations. I hope you enjoyed that, and I'll see you again next time. Hello, and welcome back to Mr. Rowe Science. Today I'm going to talk you through the first part